Hi everyone, welcome to Physics of Behavior. It's nice to see you. I invite you all to take a nice deep sigh of relief. We're here, we made it. In the course, we are inside the second, third, the middle of the course. So the course is divided according to principles. As I said, the first third of the course was devoted to principle of network motifs. And we saw that complex biological networks can be broken down into simple circuits. They happen all again and again and again. Evolution converged on them because they carry out specific functions and they can be combined in ways that preserve their functions. And now we're in the second third of the course, which is devoted to principle of robustness. Robustness is the idea that biological circuits have a special design. Let's take a nice deep double sigh of relief. So for you. And this design makes the essential feature, the essential computational feature of the circuit insensitive to the naturally occurring variations. For example, in the concentrations of the proteins that make up the circuit. So how can you have a precise computation with noisy components? It is insensitive to the natural variation. So we'll always have to ask, what feature is robust with respect to what variation, what fluctuation? And we talked last time even that other kinds of robustness, that our cells, nice deep sigh of relief, our cells in our body need to be robust to mutations, because in our body 10 to the 14 cells, 10 to the 14 divisions, and cells keep dividing. If there's a mutation that makes your receptor active all the time, telling the cell to divide, that's a step towards cancer, right? So cells need to be robust in our body to mutations, for example. A bacteria, that's not such a big deal. If there's a mutation, that bacteria will be selected against. That's not a big deal, robustness to mutations. But bacteria, and also our cells, need to worry about changes in protein level, because you can't control protein levels in biological material very well. So how can you have a precise computation of proteins made out of circuits made out of proteins when the protein concentrations in each cell are different? That is the question. And we'll see that, as we saw last time, the need for robustness makes biological circuits sometimes take on very elegant, special structures that make things cancel out so that you have robustness. And I know that some of you were on a retreat last week. How many of you were in this complex system retreat? So I wanted to make a review of what we did last time, and for everyone else also. And last time we talked about robustness. And we also kind of shifted gears and instead of talking about transcription circuits that we did in the first part, which are slow, remember they take hours to make new proteins, we focus here in this part on faster computations the cell makes with already pre-existing proteins. And those proteins modify each other chemically, and this therefore can make computations on the scale of seconds, because you don't need to make new proteins. These are called signal transduction networks. And the way they're built typically is that there's a signal. A lot of times it's a molecule, like let's say a hormone that tells the cell to divide in our body. Yeah? And this fits into a receptor. And the receptor is a sensor. Part of it is outside of the cell and part of it inside the cell. And when the signal binds, the receptor changes conformation, and inside the cell acts as an enzyme that can now add a bit of information to a messenger protein that can diffuse in the cell, go to the genes, activate them. So this messenger protein is modified, and the typical modification is called phosphorylation, adding a phosphoryl group, PO4 minus. And the rate that this happens depends on the signal. So the receptor can sense the signal and changes the rate that this modification happens. And then there's another protein Z that removes the modification. So phosphatase kinase there. And this is the output of the system. It goes to the genes and makes the proteins in response to the signal. For example, if this is a hormone telling the cell to divide, these genes will make up the machinery that makes the cell replicate its DNA, align the chromosomes, divide into two. And this happens here on the time scale of seconds. And we noticed that this circuit, what it does, this is the input S signal, and this is the output Y phosphate. And we can think about the input-output relationship. So for each input, the more S there is, the more YP there is. This is the input-output relationship. And what we notice is that we solved this circuit, and we notice that this circuit, if we change protein levels, like so if we have more receptor or more of this Y, then this input-output relationship is not robust. So all this works on seconds. Having more X, Y, more Z is a property of that lasts for entire cell generation because protein levels are made by transcription networks. So if you happen to have more X and more Y and you have a certain signal, this cell with more X will have a different output than this cell. And each cell will have a different output, and that's fine in many cases. Like we'll see today a case where that's good actually, because you can have diversity in response. But in some circuits, you want precision. For example, circuits that sense osmotic pressure, you don't want to misread the osmotic pressure and do something that will make the cell explode or shrink. You want precision. So the question is, so this is a non-robust. So what is non-robust? The input-output relation here is not robust with respect to variation in the proteins that make up the circuit. Did I explain myself here? That's the property, input-output relationship, is not robust with respect to this variation. Any questions so far?
So the VS, does it actually depend, the rate does actually depend for a single enzyme on the amount of S, or it's just the average over many X's that this is the interaction?
Yeah. So this V of S, where it comes from, what it is, it does depend on a single X, etc. So this depends on a single receptor, basically. It's the receptor switches active and inactive conformations on time scale of microseconds. When S binds, it says bound for height. Nice deep sigh of relief. Welcome. It says bind for like a millisecond. So on that time scale, it changes the free energies of the active and inactive states so that the activity now changes, the average over this. So that V of S is the average over time of the binding unbinding of these conformations, which happen on a millisecond time scale. And therefore, it's a single molecular, it depends on a single molecule like this.
So the concentration of S affects it through the time it takes to reconfirm because it binds and unbinds?
Yeah, usually you can think of free energies. So the active and inactive state has certain free energy, and temperature makes it flip like that. And when you add S, it can stabilize, let's say, the active conformation. And now the probability of being an active conformation is higher. But since these are microsecond transitions, and we're talking here about seconds, you can think of the probability to be, or actually the rate, the number of times per second makes the first conformation depends on S in this way.
Depends on the
Concentration of S through the free effect on free energies. It makes like the free energy well of the active state more deeper. Or another receptor may make the not inactive state deeper, and then S actually inhibits the binding. So it depends on what you want to achieve. Did I explain myself? This, and then we asked, is it possible, in principle even, to have an input-output relationship where no matter what is changes, nice deep sigh of relief, welcome, in the proteins, do not affect at all the in-out relationship. So this will be a precise reading of the output without depends on the proteins. Yeah.
When you say robustness, do you mean that a big variance in the signal, in the protein levels, Y0 and YP,
Yeah.
Still result in the same amount of protein.
So
It's the opposite of sensitivity.
it's the opposite of sensitivity to protein levels. Yeah. For example, if I have more receptors per cell, it's only natural that a given signal will give me more YP, phosphate. Also, if I have more total Y, it's only natural that I'll get more signal. So that's why most circuits behave like this, more YP. But can you get this, where I add now, a double number of receptors, or I double the number of Y, and I get the same YP? Something has to compensate for it. That's why I was thinking this can't be, but it turns out that bacteria solve this problem, and there's thousands of examples of what's called two-component systems. This is just a review, right? And they work like this. They have similar components, but there's like additional details that appear to be meaningless, but they give robustness. So first of all, X first phosphorylates itself. So it takes ATP, and it takes the phosphoryl and phosphorylates itself. And then it transfers it to Y. In fact, it's this ATP-bound form here that takes the ATP, the P from ATP, connects to itself, makes it. And then this phosphatase, the reverse step, is carried out by the same X unphosphorylated bound to ATP. So this is a bifunctional prot component. It does two things. It both adds and removes the phosphoryl. And this is not just a reaction in its reverse. It's actually two different reactions on two different sites. Yeah.
I decided I found that weird because X is sort of bound to the surface and Y is floating around there. Maybe it went to the nucleus. Why would it ever lose its phosphorylation if it needs to get back to the membrane in order to do that?
Right. So the question is, why diffuses? Why would it ever go back to the membrane? So in bacteria, remember, there's no nucleus. It's all one cytoplasm. And the proteins, in about 10 milliseconds, they diffuse across the cell. So think of this YP. They're all blah, blah, blah. So they find the receptor many times a second.
They get far away from the membrane and never see it again.
No. And also in human cells, instead of taking human cells, is 10 times bigger, so it takes 100 times slower. So instead of 10 milliseconds, it takes like a second to diffuse. Sometimes they also have fibers with motors that carry them. So there's a spatial component. Sometimes it's important in big cells, but not in bacteria so much. I mean, of course, there are exceptions.
So in regard to the previous question, what happens when the signal is released and acquired again, and the X already is XP? So does it lose its spin when it goes back, or it remains XP?
This reaction here is very slow because the cell makes sure to make much more ATP than ADP, and therefore this almost never happens.
So even if the signal disappears, it's still on
Yeah,
X.
yeah. And then Y0 comes and takes it. So all this takes seconds. Yeah, good. The upshot here, you remember we did this, this took us a while to compute. This turns out to be very simple. We looked at the box, like this whole thing like a black box, and we asked, when does these phosphoryl groups, when do they go in and where do they go out? And these have to be balanced. They go in from ATP, get tied into the system here at the rate V of S times X0, because each X0 has a probability V of S to time it. And they go out only in one reaction, which is here, which is the rate of this reaction, let's call this V2, is the same thing. It's V2 times X0 times colliding with its substrate, Y-phosphate.
Times XP.
Times X0. It's the same X0 unphosphorylated. It does. Yeah. And if you want, I can even put in here ATP, because it's X0 bound to ATP, if you want. Okay. There's an extra bonus for you in this review that I'm doing of last time. This special bonus, because it turns out...
It's down there.
What?
Why do you have ATP there?
Here? Because the same ATP bound form here. This is now I'm adding some information.
So when it takes it, when it moves YP to Y0, it
No, it doesn't need it, but it does need it. It needs it as, this is again one of these biochemical details people know about but couldn't explain, is that to take it away, it needs to be bound to ATP, but it doesn't use up ATP. And now we'll see that it's needed mathematically for this beautiful cancellation. This has to be equal to this, otherwise phosphoryl accumulates. So the influx and outflux at steady state have to be equal. Otherwise phosphoryl goes up, 1 second. And therefore, we can cancel out the things that happen on both sides. And we get the answer that the output is just the ratio of these molecular rates. No, there's no X or Y proteins here, levels. And therefore, we achieve this absolute concentration robustness. Why does this work? It's because if there's more X, there's more of this, and both of these reactions are speeded up, but the ratio remains the same. So having the same X do both things, if we have more of it, both reactions are speeded up, but the ratio remains the same. And also, if we have more Y, then it changes here the ratio between X0 and XP that can compensate for it. More Y takes away more of this XP, so there's less XP, but the system adjusts to the same steady state. And it's basically this cancellation is kind of like a little miracle built into the special design of this circuit with this bifunctional component that does two different things in the circuit at the same time. As I said, this circuit occurs 30 times in E. coli and thousands of times, known times in the thousands of bacterial species that have been sequenced and studied. So it's like a recurring module for a robust input-output relation. And that's kind of an introduction to this idea of robustness. It's special structure, usually has simple mathematical solution too. Nature is kind to us in this way and gives us this precision. Yeah.
I think more X will give you more redundancy in the cell, but the downside will be that it will consume more energy.
So why more receptors? Why have more receptors? What's the trade-offs that determine the number of receptors? Because
like redundancy versus
so truly, if this input-output relationship doesn't depend at all on the number of receptors, why not just save energy and move them down to very few? So that's what the cell actually does. There's very few, but you can't go lower because then it becomes stochastic. So if you have one, some cells will have 0, some cells will have 2, and 0 cells can't sense. So you need to be a few standard deviations away from 0 to have some sense. It also affects the response time. This is just a steady state. And why? Usually there's a lot of it, and the reason has to do with the problem of finding its site on the DNA and things like that. So its proper site on the DNA, and it usually has many target genes. So you need a lot of Y to bind all those target genes at once. So this osmotic response, there's several transporters that need to be changed, enzymatic pathways that change the osmolarity inside the cell, telling the cell, we're probably moved from the water outside to inside the body because there's high osmotic pressure. And if there's happening to be also low iron, low oxygen, we're probably inside the intestine. Okay, we have to switch clans. Now we're inside the person. Now we need to deal with the immune system, the thousands of other bacterial species in there. So this is part of the sensing. So there's many genes actually regulated here. And then there's 30 other sensors for magnesium, for phosphate, for the two-component systems. And altogether, E. coli builds an accurate picture of its world. Thanks to this robustness, it knows quite precisely how much magnesium, phosphate, etc. there's outside. And then it can make decisions that are critical to its existence. Am I in the upper intestine, lower intestine? It's very interesting decision making. This was the review. I want to move on, just like a bacterium with the flagella, moving on. So I'm going to erase. So I'm going to talk about bacterial chemotaxis. So I drew these bacteria there, moving. Yeah, I'm going to tell you. Building up some suspense. When you take bacteria in a dish, they're swimming, and you put in a little pipette with something they like some amino acid aspartate. They swim to the pipette. If you put a pipette that has something like nickel ions or amino acid leucine or something, they swim away. What I'm saying is, if you have a gradient of certain chemicals that they like, they call them attractants, the bacterial population moves up the gradient. And it moves down the gradient of repellents, of things they don't like. That's called chemotaxis. Chemo for chemical, taxi for calling a taxi to move, right? Chemotaxis. And this is an amazing ability because these bacteria are facing tremendous physical challenges because they're so small that water molecules hitting them cause Brownian motion. So if they try to swim for 10 seconds in a certain direction, they find themselves 90 degrees to where they wanted to go on average. So they need to navigate in a storm, you can say. Plus, they can do this chemotaxis over 5 orders of magnitude of background. They can detect a gradient as small as 1 molecule per cell length, 1 molecule difference per micron gradient, over a background of 1,000 molecules per cell volume. So what does that mean? They can detect a difference of 1 molecule between their 2 ends, where around them there's 1,000 molecules. So that's their ability. I want to say chemotaxis is E. coli can chemotax to several chemicals. There's other bacteria that can chemotaxis to light, to electric fields, to magnetic fields, bacteria in gold mines, to gold. So this is like a module. You can hook up different sensors to chemotax to different things. How do they do it? The naive thought at first is that they have maybe an antenna on the front and the back, and they can detect this difference, right? But that doesn't work because, again, of the physical reasons. So if they had an antenna on the front and the back, they can detect a gradient of 1 molecule per cell length, right? So, but on the background, where there's 1,000 molecules per cell volume, so a typical sensing area, they have 1,000 molecules, and they need to detect a difference of 1 molecule. Now, if you have 1,000 molecules, the noise on the 1,000 molecules is square root of 1,000. It's every 30 molecules. So you need to detect 1 molecule when the difference between the back and front is something like 30 molecules. You can do that if you integrate over enough time, but by that time the Brownian motion already moves you, so you can't do it. So you can't, that's not the way to do it. By the way, our cells, like immune cells, they can have antenna in a certain part and integrate over minutes and move over hours like that, but not E. coli, that needs to do it in seconds. So how do they do it? And the answer was discovered in the 70s by Howard Berg. He was a physicist, and he built a device, a tracking microscope. So he could look at the bacteria swimming, and the microscope would move the stage so the bacteria is always in the center of the field. This was very difficult to do in 1970 electronics because the response time was similar to the response times here of 10.1 seconds, etc. But he could do it, and he discovered that bacteria solve this problem. Bacteria detect changes in time. And the way they do this, they do a biased random walk. So when you look at the bacteria, they swim for about a second. And then they do this kind of change direction, and they swim in another direction, they change direction, they swim in another, change direction, like this. These are called runs. The straight paths are called run, and they last about a second. And these are called tumbles, the random change in direction. They last about 0.1 seconds. So bacteria do runs and tumbles. That's what it looks like when there's no gradients around. They tumble about once per second. Now, if you put in a gradient of attractant, they do the following. If they're moving towards the gradient, as if concentration is going up with time, if things are getting better, they reduce the probability per unit time of doing a tumble. So they keep going. And if they're moving in the wrong direction, let's say the attractant concentration is going down with time, things are getting worse, they increase their probability to do a tumble, and therefore move in a different direction and have a chance to go back to the right direction. Of course, they have no idea what direction is. They're only operating in an abstract space of concentration and time. So I'll write that again. Their strategy is, if things improve, that's to say the concentration rises of attractant, then reduce your tumbling frequency. Tumbling frequency means how many times per second you tumble. So if things are improving, keep going. Keep going. If things get worse, concentration is going down, then increase tumbling frequency to change direction randomly. So this little algorithm, in fact, guarantees you, on average, to move you in the right direction. So questions?
The definition of improve, based on last time or on some
So question,
bigger memory?
when I say improved, concentration rises, you need some memory, right? You need to compare now to the past.
To the one last.
So we'll talk about the circuit. How to do it? It's basically a temporal derivative computed with a protein circuit. And a logarithmic derivative at that. So it's like the questions, yeah.
So the sensitivity is integrated over time? It's an average sensitivity to one molecule per cell length? Or is this because they only stop to measure every once in a while?
Yeah, so you see, they don't stop to measure. They continuously sample along a random walk. So their concentration at their point, it looks like, so there's a very noisy signal. So with where they're going, where the gradient is noise,
So they integrate over time?
they need to integrate over time. Yeah, so we'll see how they do that. But they can't integrate for very long times because of this Brownian motion, so they have a limitation.
So on average, the frequency of these tumbles is still, I mean, it's regulated enough. Obviously, this Brownian motion would always cause them to change direction.
Yeah.
So how can they maintain the path, say, they detect that things improve, and yet they will always be carried away?
Yeah. So the question is, how do we include Brownian motion into this picture? So if the runs are 1 second, Brownian motion isn't very strong, but if they're 10 seconds, then it's very strong. Did I answer your question?
Yeah.
How
Yes.
do they actually change direction randomly? How do they vary the tumbling?
How did they change direction randomly? So I want to zoom in on the bacteria for a second, how they do this runs and tumbles, and then we'll talk about the circuit. But so right now, that's exactly what I wanted to show now. The way that it works is that if I look at this bacterium now, they have typically 6 to 10 motors, the flagellar motors I told you about, electric motors spin at 100 hertz, that's connected to the flagella. That's like a corkscrew pushing them at 30 microns per second. Now, these motors are not only electric motors spin at 100 hertz, they're also bidirectional, and they can spin clockwise or counterclockwise. When they spin counterclockwise, all of them spin counterclockwise, you get a run, because the flagella are built of proteins that have a natural helicity, and when they're spinning counterclockwise, the hydrodynamics make them bundle together and push you through. But if one motor changes to counterclockwise, to clockwise, its flagella flies out like when I do with a ponytail for my daughters this morning. I need to improve in that. And then the bacteria changes direction because you have one flagella sticking out like that. And that has to do, again, with the protein helicity encoded in the proteins that make up the flagella bundle, which itself is a polymer made out of proteins that move like this. It's hollow, and the new subunits move inside and complete at the end. So it's like an amazing architecture. So now we need to talk about what makes the proteins to go from clockwise to counterclockwise, which is inside the cell will have a little protein circuit that does it. Yeah, a cool experiment is a single molecule experiment. Actually, you can take the bacteria, cut off all the flagella by shearing it, and stay with one flagella glued to a glass surface, and look into the microscope and observe it changing directions. Then you get a signal. If this is 10 seconds, you get a signal that looks like this, going from clockwise to counterclockwise. Clockwise, counterclockwise. So you can see a single motor switching. And then when you add that, when you add that tractant, as we'll soon see, it thinks things are getting better, and it just stays counterclockwise. Yeah? So you can do it on a single molecule level, single cell. And this motor is a subject of great study and a beautiful machine. So let's see where we are. Chemotaxis, attractants, repellents, Brownian motion, no antenna, runs, tumbles clockwise, counterclockwise, the tethering experiment, and the tumbling frequency. So we did all this row here. Now let's look more at the computation that this bacteria does, and then look into the circuit, and then ask about robustness. What's robust here? I'm going to erase this part here. Bacterial chemotaxis is probably the best studied little signal transduction circuit, and it's like the hydrogen atom of signal transduction. And in it, in the system, one reason I'm teaching it is because it was the place where a lot of principles in systems biology were discovered, because it's so well worked out, because of the people who worked on it, like Howard Berg and other people I'll mention too. So, now I'm going to show you just an experiment. Actually, this experiment was an experiment I did in my postdoc in Princeton, 1997, 8, something like that. So, nice deep sigh of relief. Welcome. Thank you for chemotaxing in. So, this experiment is as follows. What you do is you take a bacteria and you let them swim. And actually, it's very simple. You take a slide, you make sure to dip it in something so the bacteria won't stick into some agarose. And you put a little drop in, and you take a grease pen and you make a circle around the grease pen, and then you put a cover slip on it, and the little drop spreads into like basically a two-dimensional bacteria gas like that. And you see the bacteria moving. And in this experiment, this is time, and this is tumbling frequency. So how many times per second they do these tumbles, right? And as I said, they do typically 1 tumble per second. This is the average over all the cells. So it's tumble once per second. Are you with me? Am I explaining myself, I should say? And now, at this point in time, we add attractant, like alpha-methyl aspartate, an attractant the bacteria can't eat or do anything with, so it stays there. No matter which direction they're swimming, they think things have just gotten better because there's a step up in attractant, right? Things are better. We're probably moving in the right direction. Yeah? So what do they do to their tumbling frequency? It goes down. Within about 0.1 seconds, it goes down. So on this time scale here, it's actually, you can't even see it like this. And it stays down. And for about a minute. But then the bacteria, somehow they're swimming, right? And they realize they've been fooled. They say, wait a minute, what's going on here? And they
realize because there's no improvement, right?
Well, there's no further improvement. And they recover back. So after a few minutes, if you look at the microscope, you can't tell that there's a lot of attractant there is, but there is, right? So there's a lot of attractant, but they can manage to subtract it out, ignore it, whatever. And they return back precisely to the same tumbling frequency. So this is called exact adaptation. Despite the increased input, the tumbling frequency, the output, tumbling frequency, returns to its baseline level. And Yeah, it's like a derivative in a certain way, because it only takes the derivative here, or if you spread it, derivative is like this here. Of course, they have more, so it's like a spread out derivative. Correct. It's like changing, derivative means change in time. It's the sense that changes in time.
They don't have any proportional.
They don't have any proportional controller because they reach back exactly to the baseline level. And proportional control always has an offset. Yeah. You know, this exact adaptation is not only in bacterial chemotaxis, it's in many sensory systems, including in our body. Like, for example, when I go into a dark room, I can't see very well, but slowly my eyes adapt, and I can see changes in contrast again. Okay, so this is, what the connection is that exact adaptation, if there are attractant, we have response and exact adaptation. If I have another step here, I'll also get response and exact adaptation. And another step here, I get response and exact adaptation. So this keeps me poised for further changes.
Saturation?
Yeah, so at some point the receptors get saturated and you don't respond anymore.
The tumbling frequency is 0, etc.
So if you have saturation means if you put in, let's say 10 millimolar of this alpha methyl aspartate, and now you add another 10 millimolar, they can't see it anymore. That's like a saturation of receptors. But if you go from 1 micromolar to 2 micromolar to 3 micromolar to 4 micromolar to 10 micromolar, 100 micromolar, 1000 micromolar, back to 800 micromolar. By the way, if you take away attractant, you get this response with exact adaptation. So taking away attractant means, oh, we have to change direction. So they tumble more. Take some more away, they get this. And that's basically the way our senses work in vision and hearing. So we have an annoying background noise, and after a while we don't hear it anymore. But we can now hear little changes. Let's see. So I have this tractor outside, now I don't hear it anymore. Now somebody turned on the music, so I can hear that, and then I don't hear it anymore, and then somebody starts talking to me, so I hear that. So also, if you have multiple signals, if you want to be sensitive to multiple changes of different things at the same time, right? So it's nice to come back to a poised level of tumbling. So that's exact adaptation. Occurs in almost all sensory systems. Not in pain, for example. Yeah.
Does the bacteria check its environment all the time, or only while tumbling?
The bacteria senses the environment all the time as it's swimming because it has receptors and it's a continuous system.
Does it drop in reverse though? If you put more attractant, will it drop lower or less?
If I add more attractant?
Yeah.
Oh, the amount it drops depends on how much attractant I put in. It's very sensitive, so as I say, it responds over 5 order of magnitude. So, in fact, very small changes in attractant, relative changes, cause a large decline. And also, the adaptation time depends on attractant. So, big attractant steps, like going from 0 to 10 millimolar, takes 10 minutes to adapt, but going from 0 to 1 micromolar can take a few seconds to adapt. So, everything adapts. So, that's the basic phenomenology we want to understand. We want to understand response, quick response, and slow adaptation, and exact adaptation. And I can ask, what's robust in the circuit? That is to say, if I change protein levels, does everything break up apart? Or do some things stay robust with respect to protein levels? Maybe give us a clue what's important here. So I'm going to ask now the question of robustness. So, okay. I'm just going to basically tell you where we're going. Where we're going is that we're going to see that most models you can build of the circuit I'll tell you about in a second, you can get exact adaptation. With certain numbers, protein levels. But if I change the protein levels, I change the steady state tumbling frequency, and I don't get exact adaptation.
What protein are you changing?
Yeah, I kind of went too fast now. I want to tell you about the proteins inside the cell. So most models are fine-tuned with respect to the property of exact adaptation, are not robust. And I want to talk about this robustness of this feature of exact adaptation. This, I want to see if we can make this robust. Yeah.
There are two kinds of, you said that they need to have a memory, right?
Yeah.
So, but this adaptation is not the time scale of the memory, right? It's the...
This time scale will turn out to be the time scale of the memory. So it's minutes if it's a big step, but in typical steps it's more like seconds. About a second in memory.
Okay. So it depends on, the memory is dependent on the change in the...
Yeah. It's very sophisticated. Okay, I'm going to now, if any questions about this experiment, you understand this experiment? I add attractant, a response, exact adaptation. Any questions about this? Yeah.
If you have change before you have the exact adaptation, it lowers the concentration. It feels
Oh, what happens if there's a change in this part?
Yeah.
So there's a response. Depends where you are. There is a response, and with the model I'll tell you about, you can compute it if you want. So in general, this is just this is like an approximation for what bacteria really sees, whereas it runs and tumbles, which looks like this, and its output is like this, and you need to make sense of this. So but this experiment is so simple. It's like it's a nice way to kind of get what's going on from this dynamical system. Okay, so let's now look inside the bacteria. I'm going to zoom in, so take this bacteria here and make it even bigger, and see what proteins inside can do this computation. Yeah.
Can I ask one question?
Yeah.
So the tumbling isn't necessarily maximal when it happens. Like it looks like from that second diagram that it can get more than it
Yeah.
actually was to.
If it's 1 per second, its steady. Its baseline is 1 per second. It can go here to, let's say, 3 times per second, and here to once every 10 seconds. So it has a range.
Why wouldn't the bacteria use its maximal tumbling?
If you're maximally tumbling, the way it looks, there's mutants like that. Maximally tumbling is just staying in place, and you don't get anywhere. And you basically really can't do chemotaxis because you have no sense of it. So the maximum the 1 per second is kind of an optimal scan, kind of. And then once in 10 seconds is already too long, basically, because you're Brownian motion. Yeah. Tumbling has is really a random walk. It's not a directed random walk. And tumbling actually all the time has an advantage where you're stuck in a gel or in a tough matrix, like maybe inside in the colon. And then the way to get across is just not to think too much. So if you're in a liquid, it's good to have long tumbles. So we're going to tell you something. The bacteria have individuality. Some of them identical to the bacteria. Some of them tumble more than others, because I think they're unprepared for exactly how many obstacles they'll see. So if you get an obstacle every second or every 10 seconds or every 0.1 seconds, your optimal search needs to be different. So that's...
This variation is just a variation in the population, in some sense that enables them to confront different environments, or I didn't understand.
I'll talk about individuality in a little bit. Okay, so let's take a nice deep breath now. We're going to dive into the bacterium. Are you ready?
Yes.
To go into the bacterium, to see the circuit, the beautiful chemotaxis circuit. So I'm going to go into the bacterium. This is the membrane now. Okay? So just like I showed you in the review or last lecture, this works with a single signal transduction network and phosphorylation. So there's a receptor, X, that senses the attractant, S. Just like in the lab I told you before. Okay, so E. coli has, let's say, 10,000 receptors like that for serine, and another few thousand for aspartate, and there's 5 kinds of receptors, 5 senses of smell. Each one actually is sensitive to several attractants and repellents, each sense of smell like that. So E. coli with these 5 receptors can smell dozens of things. Okay? But they all converge on one signal processing circuit. Inside the cell, X phosphorylates Y, just like I showed you before, right? And there's in this case, there is a special phosphatase, Z, called key Z, that removes this phosphorylation so that you can reset the system. YP, so this is the input. YP is basically the output, because this is protein diffused in the cell. Instead of going to genes and activating gene expression, in this system, it goes to the motor and makes it turn clockwise, and there increase the probability that it turns clockwise, and therefore get tumbles. So the more YP there is, the more changes of direction. So this will be.
Default direction, and each tumble is by one changing its direction.
So
Change back.
yeah. So imagine they have no idea where they're heading. So they're swimming, and when
They're all spinning in the same direction.
the motor spins in the same direction, so the bundle is right behind, pushing the bacteria forward. Now, one motor turns clockwise, its flagella sticks out, and it's basically a hydrodynamic process where, and there's almost no correlation between its previous direction and when it ends up.
But it will return to spin counterclockwise.
Yeah, it will turn to spin. It will return to the right orientation for running.
Everyone is in the same direction, and they shift.
Yeah, shift once in a while, 1, 2 shifts. Yeah. And this key Y phosphate is diffusing, binds to one of those motors, and increases the probability. This is a molecular machine again. Changes the free energy towards the clockwise direction, and increases the probability that the motor will spin clockwise. And when it spins clockwise for about 1/10 of a second, and then goes back. So this is all inside this machinery here, this motor. Okay, so this will be like our tumbling frequency. This will be our output. So we have input, output. And you can already tell that the tumbling frequency is not going to be robust here, because this is the non-robust circuit. And indeed, as I said, different bacteria, some swim higher, some lower tumbling frequency. That's not going to be the point here. But if this was all, or let's see if we can understand it. Oh, I didn't tell you one very important thing. This attractant input, okay, to understand this circuit, I'm warning you in advance, there are going to be a lot of afuch al afuch. Okay, so, but we will make it. Okay, so if you feel like there's a balloon expanding in your brain, you're not the only one. The attractant, do you remember I told you about this? Changes the rate of this addition. Attractant lowers the activity of X. It lowers V of S. So basically, this is a signal. This is V of S. It goes down like this. And there's kind of a halfway point here, K, where it goes down 50%. It looks a lot of times like 1 plus S over K to the power N. And this N comes from, as I said, there were 6 receptors, and they help each other bind ligands. So there's this kind of nonlinearity, cooperativity here. But this is the kind of function. So the bottom line is that I add attractant. It changes conformation of X to do this less, and therefore have less tumbles. So if they're going in the right direction, the more attractant there is, the fewer tumbles there are. Here there's no memory yet. It's just an input-output system. It's not the end of the story. Okay. So if this was it, so you see, I add attractant, and tumbling frequency goes down.
Low enough that they won't all spin in the other direction, and then thus again not tumble but run just in different directions.
So you're asking if all motors spin clockwise, do they run?
Yeah.
I don't think so, because there's a problem with the flagella polymer itself has helicity that interacts with the hydrodynamics so that it's different clockwise and counterclockwise. And that's because proteins themselves have helicity, because amino acids have a left and right form, and all of biology is made of one form, amazingly, and so you can build things that have chirality. It's a lot of incredible things. Look, so this is the input-output relationship circuit, but if this was all, as long as there's S outside, I would keep like this, right? Because just a reading, this takes 0.1 second. 0.1 second to put attractant. This stops, Z dephosphorylates. there's less YP, less more. All this thing takes about a tenth of a second. So this is this response time here, a tenth of a second, and that's what this part of the circuit does. So that's we know from last lecture. This is not robust. This is not has no memory. This is just an input-output relation. But chemotaxis needs to do something more sophisticated, which is to measure a derivative to have memory and to adapt. So how does adaptation work? How does adaptation work? Adaptation works in the following way. Here's a receptor stuck in the membrane, phosphorylating key Y. This is key Z, dephosphorylating it. The receptor has buttons. These buttons are yet another kind of modification. They're methylation. So here, instead of phosphoryl groups, it's CH3 groups. You can put it in 4 or 5 different spots on the protein. And what they do, it's like a gain control. These methylations increase the activity of the receptor. So these methylations, the more methylated it is, the more it makes tumbles. So the more methylation, the more active is X. So let's say X star or V of S is higher. Right? And the more tumbles. Now, these methylations are again added and removed by dedicated proteins. So they're added by a protein called KR. So this adds, takes S-adenosine, takes the methionine, puts it on the receptor, and it's removed by key B. So this chemotaxis system has receptor, key Y, key Z, key R, key B. That's the circuit. And they're all sitting, by the way, on one operon. R, B, Y, Z. Key R, B, Y. All right, and so this R is a very slow enzyme, and there's a few R, and it's slow, and it basically works at a constant rate, and just keeps adding methyl groups. It always has, almost always has, a substrate and a little place to put a methyl group. So this is a nice, constant influx of methyl groups all the time, adding methyl groups to some receptor.
It is completely independent of the activity of binding of S or
Yeah.
whatever.
But B is where, so we need a feedback loop here. We need to know that S is bound so that we can adjust the methylation, right? And this feedback loop is that the more active X is, the faster, you can say B works. So to reduce methylation, because it takes it away, and therefore to reduce tumbling. So if X is active, there's more tumbles. B becomes more active, and methylation goes down, and X becomes less active. So this is a negative feedback loop. I'm going to explain it in a second again. This again is reduce tumbling. So what we have here, and another important thing to say is this part here, all this part here, R and B part is slow. This takes minutes to completely methylate and demethylate. So the response is 0.1 seconds. This adding to completely methylate all the receptors is minutes because R works so slowly. So that's the separation of time scales for the response and the adaptation. So the way this works is the following. So if I put in S, add attractant, I look here at activity of the receptors, which is like tumbling. And it goes down because of that part within 1/10 of a second. And here I'm going to talk about this methylation. It starts rising slowly, 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 slowly. Why does it rise? Because X drops, therefore B is less active, therefore less removal, therefore only accumulation. And this causes activity to go back.